for this. Okay, so there are a couple of different ways in which marine microalgae as well, aquatic microalgae do this as well. They have strategies to enhance the amount of CO2 within the cell. So we know that CO2 in aqueous solutions is in equilibrium with bicarbonate. In order to enhance the flux of inorganic carbon, some plants make an enzyme that's carbonic anhydrase. Carbonic anhydrase simply increases the rate of exchange between CO2 and bicarbonate. It doesn't cheat thermodynamics, but it cheats kinetics. So it just speeds up this reaction. If you speed up that reaction, particularly right at the right very near the cell wall, you can increase the concentration of CO2 right at the cell wall. Other plants take that bicarbonate directly in. It's a charged species, and it can kick out some protons and suck up that bicarbonate. There are other plants which, in a mechanism that I don't really understand, that they can take molecular CO2 and enhance the flux of that CO2 into the cell. It takes energy to pump out protons too. Here's some work that demonstrates the uptake of bicarbonate in some plants in the Southern Ocean. This is work of Nicholas Kassar, who's a PhD student in Hawaii, who's now a faculty member at Duke. Nicholas really wanted to go onto a cruise in the Southern Ocean. It was the Southern Ocean Iron Experiment. Did you hear about these iron experiments? There are these regions of the ocean that are called high nutrient, low chlorophyll. There's lots of nutrients, nitrogen and nitrate and phosphate present, but the chlorophyll is much lower than you would anticipate. So plant growth is limited by something, and iron is one possible explanation. So what happened with the Southern Ocean Iron Experiment, there were two ships. One of them just took iron sulfate, and dumped it in the ocean. It kept dumping this iron in the ocean, and it produced a bloom. And so the experiment was to follow the fate of that bloom. Did it produce more organic matter, plant organic matter? Did production increase? And was it exported? Well, somewhat equivocal about whether it was exported or not. The grazers went crazy and they cropped it down for the most part. So what motivation is, can we take iron, I don't know, old rusty cars, dump them in the ocean and produce blooms that will sequester CO2 from the atmosphere and bury it either in the deep ocean or sediments? Well, it didn't quite work. But Nicholas wanted to go on this cruise. And to do it, it was an exciting cruise. Okay. He said, all right, I want to know whether Southern Ocean diatoms utilize bicarbonate. And this is a region of the ocean where, growth rates in the cold are low. So the question that you asked earlier, are the growth rates of these two species the same? Well, growth rates are low and CO2 concentrations are high. Optimum conditions for marine microalgae that would grow on CO2. Well, what Nicholas did to test this was he collected water samples, so natural populations of marine microalgae, mostly diatoms, and he gently filtered these and took the diatoms and gently put them into a little beaker so that it was a kind of a cell concentrate. And he added 14C labeled inorganic carbon, but he did it in a tricky fashion. He pulled up some of this 14C labeled bicarbonate, and then he pulled up a little bit of acid and kind of shook the syringe, so it drove all of the CO2 activity out of that bicarbonate into aqueous CO2. Now, as soon as you put that into the beaker, it's seawater pH, and you don't add very much of this 14C labeled bicarbonate, it immediately begins to equilibrate, but the equilibration takes time because the water is cold. So he took advantage of that equilibration time, the relatively long equilibration time, and that's what we see here. This is the uptake of bicarbonate and aqueous CO2, C14, into the plant organic matter as a function of time. And so the first activity should be CO2 uptake, because that's where the activity of CO2 is. And then as it equilibrates, eventually it's CO2 and bicarbonate. And that's what we have here. So you can kind of model it. This is mostly CO2 uptake. This is CO2 and bicarbonate uptake. And I'll spare you the mathematics of the modeling, but what he found was that about half of the inorganic carbon that was taken up was in the form of bicarbonate, not CO2. So plants in the ocean can utilize bicarbonate, and it's not universal across all species. Some do it more than others. Okay, here's another, these are laboratory experiments from a group in the UK. And they grew a haptophyte in culture, and they stuck a probe in from a membrane inlet mass spectrometer. Do you know what membrane inlet mass spectrometers are? Well, there's a membrane that acts as an inlet, as you might anticipate. That membrane allows gases to pass across it, and then sends it into a mass spectrometer. It tells you how much of that gas is present. So what they were doing is measuring carbon dioxide concentrations and oxygen concentrations. And they're not very well calibrated, but this is, and they drift a bit. You can see there's a baseline drift here. But this is approximately 1 micromole per liter of CO2 change, and that's about 20 micromole per liter oxygen change. Okay, so they kept switching the lights on and off. You switch the lights on and off, you switch them on, guess what happens? Photosynthesis occurs. What would you expect? CO2 would be taken up and oxygen would be produced. If you turn off the lights, well, stop CO2 uptake, and you begin to respire, so oxygen concentrations should decrease during that respiration. So that's what this is. This is dark. CO2 concentrations go up because it's not being taken up by the plant. Light goes on, boom. Here's CO2 going in. Here's oxygen dropping during respiration. Here's oxygen produced during photosynthesis. Then they did something odd. They took this inhibitor, this chemical IAC that inhibits the Calvin cycle, and they turned the lights on. And okay, here's the lights going on. There's no oxygen produced. Well, that makes sense. You've inhibited oxygen production. There's no more photosynthesis, no more Calvin cycle. But CO2 decreased in the water. It's like, kind of scratch your head and go, well, it's not being fixed. So how is CO2 going into the cell out of the water? Well, it turns out these things have an ability to take up CO2 and pump it across the membrane. It's the only thing that makes sense, because you've shut down the internal demand for CO2, yet CO2 drops. It's dark, releases it again, light, sucks it up again. So here is evidence that there is a CO2 pump in these haptophytes. A comparison of treated and untreated shows about 70% uptake and about 30% fixation. Now, some of that uptake is going to be lost through diffusion or flux out of the cell, and some of it will be taken up. But it's about 30% of what's fixed. But it's taking up a lot and losing a lot, too. Okay, they proposed that this mechanism was related to photosynthetic photon flux density. That's a fancy way of saying it's related to light. The energy comes from light. Well, that makes sense, too. In this case, they added another inhibitor that stopped uptake, and that stopped uptake by shutting down the electron transport chain. And what that did is it shut down the uptake of CO2. So again, this is something that's driven by light. The other evidence that's shown here that it's driven by light is they adjusted the light level. Here's 50 micro Einstein per meter squared per day. Here's 15, here's 1,000. And the rate of this uptake, presumably due to this purported CO2 pump, changes. It's more enhanced when the light goes up. This is important because it suggests that not only can light limit photosynthesis in the ocean by not giving enough energy for the Calvin cycle, but it can also limit the rate at which CO2 moves into the cell, is pulled into the cell in some plant species. And we'll come back to that, look at some evidence for that and how it affects isotope fractionation. So in a nutshell, that's sort of a primer on the controls on carbon isotopic fractionation in marine microalgae. So like the Farquhar and others model, it's a flux in versus a flux out. But the flux in and flux out are controlled by quite different mechanisms. There are no stomata in marine plants. There's no need for it. They're bathed in water, so they don't have to worry about water loss, right? But surface area to volume can control CO2 flux. Acquisition of CO2 through mechanisms by which CO2 is pulled into the cell can also control carbon isotope fractionation. So how are we going to be able to use this to understand CO2 variations through time? I'll talk about that in a moment. Any questions about this? Pretty straightforward? Yeah.
So they have this active CO2 pump, but they're only fixing 30%?
Yeah.
So what's going, like, where's the rest of the CO2? It just goes back up?
Fluxes out, yeah. Remember, if we don't have flux out, we don't have any fractionation, right? So there's always flux out if you measure fractionation. It's dependent upon what the flux is out of the cell. We'll see with same controls nitrogen isotope fractionation in marine microalgae. It's a pump system too, and that fractionation is really constant. We'll see that's really constant in the ocean. So the plants are doing something to maximize the amount of CO2, nitrogen that enters into the cell. So it's the same thing, flux in, flux out. Okay, yeah?
We didn't really talk a lot about...
Wait a minute. I guess if I was knocked out of the horseshoe round, in round one, I'd wear beads in my hair too. I'm sorry, go ahead. Oh, it's not beads, all right. I'm sorry, did you have a question?
No, I just love to get shit on.
Okay. I can give you more.
Bring it.
Bring it on, man.
I was curious about calcification.
Yeah, you know, there's CO2 that's produced during calcification, right?
Mm-hmm.
It depends on how much and how fast that calcification occurs. Under very low growth rates, it turns out that calcification or low CO2 concentrations, there is some evidence that calcification plays a role in controlling flux in, flux out in carbon isotope fractionation. Again, there's a lever there because is calcium carbonate enriched or depleted relative to CO2? Think about bond strength. Think about, the heavy isotope likes to be in the bond where it's more strongly bound. So, yeah. So you're taking an enriched source, making CO2. They produce this calcium carbonate within the cell, and then they somehow push it out. And yeah.
So they're only using 30%. Why is that advantageous to be that inefficient?
Why is it advantageous to be that inefficient? I don't know. I'm not sure. Why wouldn't they use all of it? No, that's a good question. I mean, what they're doing is that, the CO2 concentrations, even in the Southern Ocean where it's very cold, are about 40 micromole per liter. And the concentration of total dissolved inorganic carbon in the ocean is on the order of 2,200 to 2,200 micromole per liter. So if you need CO2, you develop mechanisms by which you can bring it into the cell. But it can diffuse out. You can't take up all of that. So I don't know. I think they're trying to maximize what they can get out of seawater by using this mechanism. It takes energy, so it's a trade-off. If they use too much energy and fix 100%, maybe that's not efficient enough. I'm making it up, but that's not my area of expertise. So, but it demands for fractionation to have at least some leakage. Yeah.
So does IEC inhibit photosynthesis in
It
general?
stops the Calvin cycle.
Okay.
Yeah, so it'll stop photosynthesis.
Okay, so then when they're taking in CO2, I thought they fixed it through photosynthesis. I'm not a plant person. So.
Yeah, through ribulose bisphosphate carboxylase. So that is the Calvin cycle. Yeah.
Okay, so
Yeah.
then how are they taking it up and fixing some of it if photosynthesis is inhibited?
They're not, but they're taking it up and they're not fixing it. If we look back at those graphs here, when they've added it, it's kind of tough on this because the quantification is not great. So, it kind of looks like this is drifting up there, and the baseline's kind of drifting. It looks like as much as being taken up is released when the lights are turned off. So they're taking it up. They're probably losing some of it through efflux out of the cell. But then when the lights go off, the transport mechanism stops, and so it's released back to the environment or back to the water. So,
Okay, so what is
yeah.
the 30% fixation that's happening?
This is a comparison of treated and untreated. So when they treat it, when they add the inhibitor, they can make an estimate of how much of the uptake is due to this transport mechanism. And the untreated is both transport and fixation under those conditions. Yeah?
Is there a clear life history trade-off between organisms that use mostly bicarbonate versus mostly?
I don't know. Bicarbonate, because it is a charged species, the mechanism is easier to understand. You can kick out a proton and pull that bicarbonate in. But I don't really understand the mechanism by which an uncharged species like aqueous CO2 is moved into the plant. So I don't know what the evolution of this is, or even how to test that through time, other than being able to look at sort of the genetics of it. But no, I mean, that's a good question, and it's an open area for research.